Action potential. Our body is electrically neutral, with equal amounts of positive and negative charge. Some areas are more positive or more negative than others. Because positive and negatives attract, we need membranes to separate the ions until ready for use. These are separated to build potential. It just needs an event to trigger the release. Currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membrane. A resting neuron starts off more negative. This has a resting neuron potential of usually -70. Around this cell is lots of sodium, which is positively charged. Inside the cell is positive potassium, but they are mingled with bigger negative proteins. This means the cell has an overall negative charge. For every 2 potassium ions in, it pumps 3 sodium ions out. This changes the charge of the neuron. We call this an electrochemical gradient. Large proteins which provide safe passage across the membrane are called ion channels. Voltage-gated open at certain membrane potentials, open at around -50. Ligand-gated channels open when a chemical such as serotonin is released or a hormone. Mechanically gated channels have a physically stretched membrane. The movement of ions is the key to all electrical events in neurons, thus makes up how we think and live. When a small bit of stimulus does not trigger enough to cause an action potential, this is called a graded potential. If the stimulus is too weak, for example -55, we see the voltage-gated sodium channels let lots of sodium in, which makes the cell depolarized, thus positive. This makes the action potential become very large. Repolarization makes the voltage-gated potassium ions open and take the potassium out of the cell to restore the charge. At first, too many are let out, causing hyperpolarization, -75 charge. So the sodium channels open up and restore the cell back to its normal charge. A weak stimulus tends to trigger less frequent action potentials. Action potentials vary via speed. Blood vessels and glands are slower. This relates to how much myelin is around the axon, so the signal does not diffuse. But what should always be remembered is action potential is always the same.